Bismillahirrahmanirrahim. Assalamu alaikum. Hello and welcome to Biosciences with Shatrin Sawati. And as I have discussed about the molecular targets of the SARS-CoV-2, two of them I have discussed in my previous lecture yesterday, and today I will discuss about the two other most important molecular targets of the SARS-CoV-2. So must watch my lecture, first part of that lecture, and this is the second part, and I hope so you will enjoy this lecture. You will get a lot of information from these lectures, and your basic concept about the molecular target will be clear upon you people. So in this lecture I will share some research article with you. You must go through this article because I like that article. So I'm sharing with you. You must read this and you will get a lot of information about the molecular targets. Okay, so in this lecture I will discuss about the furin and the neuropilin, as I have discussed about the ACE2 and transmembrane proteases, serine proteases in my previous lectures. So now, what are furin proteases and their role? Very important. And you can say that one of the best initiator, or you can say they're one of the best activator of the inactive spike proteins of the SARS-CoV-2, as we know that the spike proteins, S proteins, are inactive when the SARS-CoV-2 do not in contact with the host cells or human cell. So what will happen, transmembrane proteases, serine proteases activate by cleavage of the spike protein into its subunit like S1 and S2. Just like that, furin will help, furin proteases help the spike protein to break down and become the active. And how, I will show you. So first of all, what are furin basically, because most of the students are not familiar with furin. I'm here giving you the basic concept. You must go through all these molecular targets, molecules, enzymes, proteins and genes. You will get a lot, and this is very interesting, and I'm just giving you the basic concept. So furin is an enzyme that in human is encoded by the furin gene. Some proteins are inactive when they are first synthesized. So what will happen? Some proteins are inactive when they are first synthesized. Then what is the, now I am going to discuss the role of the furin, how the furin activate these inactive proteins. So, and must have section removed in order to become the active. So this furin help to remove the sections of that inactive proteins to become active. Is it clear? So it help in the cleavage of the protein. Furin cleaves these sections and activate the protein. Furin is a type 1 transmembrane protein, like the transmembrane serine proteases. This is also the transmembrane protein composed of a single peptide. Must keep it in your mind. It is just one single peptide, and this is what type 1 transmembrane protein. It was first named furin because it was in the upstream region of the oncogene known as FES. FES. So must read about the FES. I am not discussing here the FES. The gene was known as FES upstream region, and therefore the protein was named as furin. Because most of the students ask why it is called furin, ma'am. So I am giving you the concept as why it is called furin, because it is present upstreams of the FES gene. Now, I'm not here giving you the detail about the FES gene. You must read about the FES. This is an oncogene. Okay. Now, role of furin proteases. As I have discussed that furin activate the other inactive proteins by removing or by cleavage of that protein. Now, how? It has been speculated that RRAR, a unique furin cleavage site. This is the cleavage site. Now, what is RRAR? These are the symbols used for the R is used for the purine bases like adenine and guanine, and A for adenine. So this is the adenine, guanine, nitrogenous bases, a nucleotide sequence. A unique furin-like cleavage site, FCS, in the spike protein, which is absent in other lineage of the beta coronavirus, such as the SARS coronavirus, which was first discovered in 2003 in China. So it was that cleavage site is absent in the SARS coronavirus, but this is present in now the SARS coronavirus 2 family. Is responsible for its high infectivity and transmissibility. Is it clear? So this site, which is present in the spike protein, which is R square RRAR, and what is R and A is the symbol used for the nucleotide bases, or you can say the nitrogenous base, adenine, purine. R uses for purine, adenine and guanine, and A for adenine. So this is the unique cleavage site at which the furin cut the spike protein into its subunit. Like you can say that the which is spike protein which is inactive become the active by the activity of the furin proteases. So furin, like the transmembrane serine proteases, act like a scissor for the cutting or for the cleavage of the spike protein. Is it clear? Okay. Now I'm going to discuss about the neuropilin. Neuropilin 1. Now what is neuropilin 1? Another host factor for SARS-CoV-2. This new mutant meant COVID-19 could suddenly latch onto an important molecule found scattered around the outside of the human respiratory cells called the neuropilin 1. This molecule helps to transport material. Now, what is the function of neuropilin? I will show you and I will discuss. This molecule helps to transport material inside cells and deeper into the tissues. The mutation was like handing COVID-19 the key to a new door into a cell, and meant the virus could replicate in a greater number in the human airway. So this is another gate for the entry of the SARS coronavirus 2, especially the new mutant strain, which I have discussed in my previous lecture. So must watch all these lectures. You will get a lot. Okay. Now, neuropilin 1 is a membrane-bound co-receptor. This is co-receptor, like SU2. There is a receptor, and this is the co-receptor of that. To a tyrosine, now this is the co-receptor to what? Tyrosine kinase. Receptor for both vascular endothelial growth factor and semaphorin and the vascular endothelial growth factor. I'm not discussing here what is the role of the vascular endothelial growth factor, VEGF and the semaphorin. Must read about the VEGF and semaphorin. VEGF help in the formation of the blood vessels and many other activities, so must read about that, and semaphorin similarly, so read about that. But here I am just discussing that what is neuropilin and why it is helpful in the SARS coronavirus entry into the host cell. NRP1 plays versatile role in angiogenesis. Angiogenesis means formation of the blood vessels. Axon guidance, cell survival, migration, and invasion. Now these are the most important role. That's why it help in the entry of the you can say that this is acting like a door for the SARS coronavirus, because it play an important role in angiogenesis, exon guidance, cell survival, migration, and the most important which is invasion. So it help in the invasions of the SARS coronavirus into the host cell or human cell. So now in this diagram you can see this is the SARS coronavirus, and this is the first receptor, which is the most important receptor of the SARS coronavirus, ACU2. And along the ACU2 there is a transmembrane serine proteases which help in the cleavage of the spike protein into its subunit S1 and S2 and activate the spike proteins, and you can say that activate the virus. So along with the SU2 you can see here there is another neuropilin. So this neuropilin also help and guide the coronavirus, and then the coronavirus causes infection, or you can say that easily enter into the cell. In other words, you can say that neuropilin facilitate the coronavirus to enter into the cell. So this is the most important, very interesting article I'm discussing and I'm sharing with you people, that neuropilin 1 is a host factor for SARS coronavirus infection. Must go to the Google Scholar and write this article name, and then you will be able to get this article. So the spike proteins of the SARS coronavirus contain a cleavage site for the protease furin that is absent from the SARS coronavirus, old SARS coronavirus, which is spread or emerged in 2003. So now this is the statement that the neuropilin 1, which is known to bind furin cleaved substrate. Now here I'm just discussing that how the neuropilin help to activate the coronavirus. Like you see that the furin help in the cleavage of the spike protein. Now what will happen when the spike protein cleaved by the furin? Then what will happen? The proteinated SARS coronavirus infectivity NPR1 is abundantly expressed in the respiratory and olfactory epithelium, with highest expression in endothelial and epithelial cells. That the furin cleaved S1 fragments of the spike protein binds directly to cell surface NRP1. Now S1 fragment, after the cleavage of the spike protein, what will happen? Two subunit form, S1 and S2. So the S1 subunit then directly bind to the cell surface neuropilin 1 or NRP1, and blocking this interaction with a small molecule inhibitor or monoclonal antibodies. Now what will happen? We can block this. The researcher believe that if we block the NRP1, then what will happen? We can easily block the entry of the SARS coronavirus into the cell. Understanding the role of NRP1 in SARS coronavirus 2 infection may suggest potential targets for future antiviral therapies. Is it clear? So like transmembrane proteases, furin, which help in the cleavage, and then the neuropilin, what will happen? This neuropilin make the entry of the coronavirus into the host cell easy. So NRP1 is just like a gate for the door for the entry of the SARS coronavirus. So what will happen? This neuropilin actually bind to the S1 subunit of the SARS-CoV and activate this. So SARS-CoV easily enter into the host cell and causes the infection. So this is all about the Furin, proteases, and Neuropilin, and most important SU2 and transmembrane proteases. I hope so the concept will be clear upon you people. If you have any question, kindly write in the comment box. I will try my best to solve to answer your question. So thank you very much. I will be back with a new lecture, and till then, Allah Hafiz.